Hi there. Right, it is 2 o'clock, so we will make a start. Let me just move the little box on Teams. Not Teams, is it Zoom? What we're using today? So that I can see the slide. So last session, we had a question about the types of port across the membrane. So these are essentially how molecules are moving back and forth across the membrane. Because on the course content, it talks, this is directly from the course content up here, it talks about primary active transport and secondary active transport, and two types of porter, the symporter and the antiport. So we've got three different types of protein, essentially, or transporter across the membrane, and two types of transport. So what I have done is put this table together. So you can see here, uniporter, uni meaning one, so that takes one solute across. We've got a symporter, so I think it's talking about just meaning simultaneous movement, two together. So 2+, and they're going in the same direction simultaneously. An anti-porter, anti always means like in the opposite direction, doesn't it? You think clockwise and anti-clockwise. I'm not sure if my camera's back to front actually on this, so that they might have been the wrong way around for you, but you get what I mean. Clockwise, anti-clockwise. Anti means in the opposite direction. So anti-porter means opposite. Port, I don't know how good your French is, but that's the word for door. So it comes from that origin, that or gateway. So uniporter, uni meaning one, symporter, simultaneous. I think that should be an M there. I think it is correct here. Symporter, I've always thought it was sim. And antiporter, opposite direction. So that's what's going on with the transport. What I was asked about is whether it was primary or secondary active. So let's just recap what they are. So primary active transport uses ATP to move molecules. A good example is sodium potassium pump. It uses ATP to exchange 3 sodium ions out of the cell, and then 2 sodium ions go into the cell. So this is a really slow way, essentially, to change the gradient of positive. Well, it's not slow, but it could be a lot quicker, couldn't it? You know, you're going 3 steps forwards, 2 steps back, if you like. So you're moving 3 positive ions out, moving 2 positive ions back in. So you only get a net movement of 1 ion from doing that. Anyway, it works. And that's an example of the primary active transport. So you can get that with uniporters, not the sodium potassium pump, but you can get primary active transport, really ATP driven pumps with this. Symporters and antiporters, no, you don't tend to get well, I suppose antiporters you can get 2 in, 2 out with the sodium potassium pump. I think that might be an example. But yes, there's a lot going on. So, oh no, that's and secondary active as well. Does it use ATP directly, or does it use the energy stored in the gradients? So no, uniporters don't tend to use the ATP directly. Symporters and antiporters do. There are so many different channels. There's all the different molecules, so it is quite difficult keeping track of what's going on, and it's best almost to look at each one individually if you're studying it. If you're studying movement across the membrane of any particular substance and you're going into more detail and more depth, look at each protein across the membrane individually and look at what it's doing and learn for that molecule. Try not to worry too much about this at the moment. This is quite detailed and a bit above the level. Well, I suppose it's, you know, you're not going to be asked a lot about this sort of stuff at this level. Yes, you need to have a sort of background of what's going on because we are going to use movement across the membrane when we talk about nerves and muscles soon, but don't worry too much about these definitions. They're not massively important at the moment. Just it's kind of like be aware that there are different types and they do different things, but you don't need to sort of memorize them or anything like that for now. It's not something that you need to worry about just yet, but maybe next year when you get onto your courses, depending on what you're doing, you might need a little bit more detail with them. Okay, so what I'm going to do is pop up the heart stuff. That was digestive system stuff. Where is my heart stuff? Down there, that we started last session. So we started looking at the heart. We had a look at the stuff that we'd already gone over, this stuff when we were looking at the different tissue types. And then we moved on to the circulatory system. So I'm going to go from that slide and catch up to where we were last session. So the circulatory system is the blood, the vessels and the heart, everything that's circulating around our body. And we talked about the reason that we need this primarily is to get oxygen and glucose around to the cells so they can make this ATP energy that we talked about. It's really important. That's how our cells do things. They need energy for a lot of processes. So oxygen and glucose are needed to make it. The exchange surfaces of the lung and the gut, the small intestine, deliver the oxygen and the glucose that are required for this reaction. It's not really one reaction, it's lots of reactions, as we'll see. But there are other functions of the circulatory system. So not only is it taking that oxygen from the lungs to where it's needed, the cells, the tissues, it's also taking away the waste, the carbon dioxide. That's waste from that cellular respiration reaction. It's taking away urea. Remember, we talked about urea being made in the liver from the amino acids being broken down. The blood also moves heat around the body. It's really important that the core, where all our organs are, is kept really warm. It doesn't really matter if we lose the odd finger or toe to frostbite, that's not going to kill us. So there's lots of temperature regulation mechanisms in place, which we'll talk about later as well. And the blood is also circulating hormones. That's like a communication system. And we also talked a little bit about defense a few sessions ago. So loads of roles, but the prime role is getting this oxygen and glucose to the cells for respiration. That's the really key, you'll die if that doesn't happen immediately role. So we started to have a look at what was making up the circulatory system. It's the heart, kind of obvious, blood vessels, also kind of obvious, and the blood, which we already know. So this is good because we kind of know this unit. It's not very abstract like cells and tissues and things like that. It's stuff that we know. It's the heart, the veins, the arteries, and the blood. So loads easier. So we started to have a look at this diagram of the heart because it is important that you understand the circulation through this heart without getting confused. This is essentially something that you would have studied at GCSE. Obviously, if you're doing an access course, it could be quite some time since you did GCSE and you might well have forgotten it. And a lot of people do struggle with it at GCSE, but this is covered at GCSE level. So it would be assumed that you have understanding of this content. So I told you that the right-hand side of the heart is on the left-hand side of the screen, and the left-hand side of the heart is on the right-hand side of the screen. That's because it's not your heart we're labeling up, it's the patient on the operating table in front of you. So obviously they're lying down. This is the right-hand side of their heart. It's not, that's the right-hand side of the screen. They're lying down in front of you. This is the left-hand side of their heart. Okay, now, if you're not sure and you're getting all mixed up, the left-hand side of the heart is thicker. Look at that wall there. It's twice the thickness of the wall over here. Okay, so the left-hand side, double the thickness. And the reason it's thicker is because that's got to pump the blood all the way around the circulatory system, all the way down to your toes, your fingers. The right-hand side only pumps it up to the lungs. That's not very far from the heart, so it doesn't need that huge muscular wall. It doesn't need all that muscle. So the right-hand side, much thinner. Left-hand side, thick and strong. Okay, so we started to label up, and I said it was the veins in. We remember that because it says vein. The veins into the heart, and it is the arteries out. So on the right side of the heart, we have the vena cava or vena cava, and this is a huge, huge vein which goes from the head and from the bottom of the body. So head and arms, actually, up here, this drains, and the whole rest of the body, this one drains. We call them superior for top and inferior for bottom. So superior vena cava, inferior vena cava. That just means top and bottom. And then we also have the pulmonary vein. Pulmonary means of the lungs. So you've probably heard of people talking about pulmonary disease. It's something that gets spoken about with regards to smoking, asbestos, mining, you know. So that's pulmonary disease. Well, they are pulmonary diseases. Similarly, pulmonary vein means the vein from the lungs. So I think that's pretty much where we got up to. We talked about the fact that there are chambers. There are 4 chambers. The top are called the atrium, or atria, actually, plural, but there is the right atrium and the left atrium, the atria, plural. So these are the chambers that receive the blood from the veins. So they fill up with blood. Then we have the valves on either side of the heart. These stop the blood from getting, when it gets into this bottom chamber and the heart pumps, these valves make sure that it doesn't go back into the atria. So this one's called the tricuspid. It's because we're going to have a look at a little video in a minute. It makes it a bit easier when you can see this moving. The tricuspid valve has got 3 flaps that you can see on the video, and the bicuspid has got 2. So tri for 3. You know, whatever language you speak, you know that in Welsh it goes un dau tri. So tri is literally the word for 3 in Welsh. And bicuspid valve, bi for 2. So that and that do the same job, but on the different sides of the heart, and they just look slightly different number of flaps, and they stop the blood going back from these chambers into the atria, because that heart is going to pump, and we want the blood to not go back where it came from. We want it to go out through these arteries, and then out up through this artery, which comes around there and there. So those bottom chambers are called the left ventricle. Now, some people find it hard to remember which way up the atria and the ventricle are. Remember it like this: V for ventricle, and the bottom of the heart is V-shaped. So V for ventricle. We've got the right ventricle, thin wall, only pumping up to the lungs. Left ventricle, thick wall, pumping all around the body. Don't forget, we got this septum here, dividing the heart in 2, because the heart has got 2 loops. They don't mix. There's no crossing over of this blood. The one loop is going up to the lungs. It's got oxygen, blood in it that's lost all its oxygen, well, not all, but most of its oxygen as it goes around the body. The other loop is going around the body. It's just been to the lungs, is full of oxygen. So before, well, I suppose after, I think it's best to think of these afterwards. Once the blood has gone from the ventricle and into this artery here, and it's gone into this artery from this ventricle, we have another set of valves, and they are to stop the blood falling back into the ventricles once they're in the arteries, once the heart has pumped. So the one that takes the blood off to the lungs, or the pulmonary system, is called the pulmonary valve. And the one that takes the blood off to the aorta, or main artery that feeds the whole body, is called the aortic valve. Thankfully, we can remember those names because they're pretty obvious where they're going. And last but not least, we have the vessels that take the blood out of the heart. So the right side of the heart goes up to the lungs, the pulmonary artery, and the left side of the heart goes up the aorta, which takes the blood all around the whole body. Now, I know with biology, there's a ridiculous amount of new words. There's loads on here, probably vena cava, tricuspid, bicuspid, ventricle, atrium, pulmonary even. There's loads of new valves, new words there. So what I would suggest you do is learn these, learn them like you would if you had a spelling test in primary school, you know, when you used to just sit down and learn to spell the words off by heart. These diagrams, I would learn to label them because you need to be able to get your heads around, need to get your head around them. If you're doing physiology courses later on, they would expect you to have understanding and be able to label this. So I would spend some time to learn these diagrams. Unfortunately, I know it's a lot of work and it's not fun or easy, but they're kind of important. I'd probably also suggest that you have a notebook when you're in these sessions and note down all the new words and write a definition next to them as well, because there are so many in biology. It's like learning a new language. Okay, so as I said, we've got two separate pumping systems operating within this one organ. The right pump is sending blood to the lungs only, and the left pump is sending it to the whole rest of the body. So the left pump has obviously got the blood that's full of oxygen. The deoxygenated blood, which has just been around the body, so it's got no oxygen left, it's been to the tissues, all of the cells have taken the oxygen for respiration. They've filled everything back up with CO2. So the red blood cells carry a bit, the plasma carries a bit of CO2, and that will go back to the heart. That comes in through the vena cava or vena cava, because this side, the right side, is going up to the lungs so that blood can get reoxygenated. So the right atrium will fill, then the right ventricle. The ventricle will contract. It squeezes from the bottom, or otherwise, if it just squished, some of that blood would get squashed down there, but it squeezes from the bottom, pushing the blood out of this artery. Then that will go off to the lungs, which are sort of up here and behind the heart bit, and it will be oxygenated, because obviously that's what happens to the blood in the lungs. It's there to collect oxygen, and it will come back via this pulmonary vein. There's a few here that are linked together, the pulmonary vein. When it's in the left-hand side of the heart, same thing happens. The atrium fills, then the ventricle, then it will squeeze from the bottom out through this aorta and off for systemic circulation around the rest of the body. And then once it's in the aorta and the pulmonary artery, these valves stop it getting sent back as well. So once it's out, it should continue on its journey. The oxygenated blood delivered through the pulmonary veins to the left atrium flows into the ventricle while it is relaxing. So you can see here, it's relaxing. It's in diastole, so it's filling up, and then it contracts systole, and while it's doing that, the blood is then sent off around the body, if it's in the left-hand side, or around to the lungs if it's in the right-hand side. When it contracts and squeezes from, you can see here, it's really clearly squeezing from the bottom up, the ventricles, and you can see in this diagram, those valves have closed while it's squeezed, so the blood can't get back into the atria here and here during systole. So diastole, it's relaxing, the valves open, it's filling up. Systole, it squeezes, and the blood goes off either to the lungs if it's on the right-hand side, or to the body if it's on the left-hand side here. Now, what you'll notice on this heart is part of it is drawn in blue and the other part of it is drawn in red. That's to show you the blood. We tend to draw on biology diagrams oxygenated blood in red and deoxygenated blood in blue. Now, it is different colors in the body. It's not blue. Obviously, well, maybe not obviously, but it is not blue, but they are different shades of red. The deoxygenated blood that we draw in blue on diagrams is the sort of color of red wine, you know, burgundy, deep red. And the oxygenated blood is more orangey red, not quite as orange as this, but more like tomato ketchup red, bright red. So they are different colors if there is oxygen in the blood. So the oxygenated blood, much more bright red. So ventricle contracts here. This forces the blood out into the arteries, the aorta up here, this big vessel here, and the pulmonary artery here, which goes to the lungs. And as I said, these valves are closed to prevent backflow. So I've just put this diagram up because it's from your course content, and it might be better for you because it's familiar, but also because I wanted to show you here where it shows the blood going into smaller vessels. So here's our heart, a simplified diagram, and you can see, let's start in systemic circulation. So this represents the cells in the body. The blood has picked up loads of carbon dioxide because of the cells reacting to get this ATP energy, and the blood is now pretty low on oxygen because the cells needed that oxygen. So it travels back to the heart. It enters from the vena cava, which is this big vessel here, into the right atrium, then moves into the right ventricle, sorry. Then it will end up going to the lungs, and you can see it moves into the capillaries in the lungs where it picks up the oxygen. The blood's now oxygenated, returns to the heart via the left atrium, then goes into the left ventricle, and then it goes off around the cells again, loses all of its oxygen and picks up CO2, and then back around the circuit it goes again. So that's what's going on there. If you have a look at this diagram, you can see the two entry points of the vena cava or vena cava quite clearly there, which I don't know, just helps you see that is actually two entry points, but they're both going into the atrium. What else can we see? Pulmonary veins, you can see the two entry points there for the pulmonary veins into that aorta, that atrium up there. Okay, so I wanted to show you this because it's because when something is moving in biology, it is much easier to watch it moving in a video than to have me talk through and wiggle the arrows. So I'm just going to put this video on. So apologies in advance if there are adverts because it is on YouTube, so they usually are. I'll try and skip through, obviously, if it lets me. Sorry about that. It's gone on to, it won't let me go on to the next slide. Right, there we go. Okay, let me just pop my questions up. How is that going through the heart? Is that okay? Did that make enough sense to you? Some people struggle a bit with that. It feels like it's a lot of information, I think, to some people. Okay, if everyone seems okay, everyone's quiet. So I'm just going to pop these up for you to have a look at. You can pop the answers up if you want to. You don't have to. You can pause the video and if you're watching and have a go at home if you're watching later, or if you're in class, have a look at these, scribble them down, what you think the answers are, and I'll pop the answers up. So what's the primary function of the heart? Which blood vessels bring oxygenated blood from the lungs to the heart? And what type of blood does the vena cava carry? Okay, I've got some answers up, so I will pop the next screen on. Well done, guys. So primary function of the heart is to get the blood circulating around the body. It's the pulmonary vein. Remember, it's vein to remember what comes in, and obviously it's coming from the lungs. That's asked in the question. So pulmonary vein, and the vena cava or vena cava is bringing back the oxygenated blood from the body to get pumped around to the lungs to get more oxygen. Okay, a little bit more thinking. This is quite hard without a diagram in front of you, really. I should have put the diagram up on the screen for you, but I'm really testing your comprehension by sticking it up without the heart. So quite challenging. In which chamber does blood from the vena cava enter? And through which valves does blood flow from the atria to the ventricles? And what prevents the backflow of blood into the ventricles? This is really tricky actually without the heart. So if you without the diagram. So if you're doing this and getting even just some of it right, that's fantastic. Okay, good work. I'll pop the answers up then. So vena cava is bringing back blood from systemic circulation to the right side of the heart. So it goes in through the right atrium, and it is the bi and tricuspid valves that separate the ventricles from the atria. What prevents the backflow of blood into the ventricles is the pulmonary and aortic valves, because that's where the blood is going from the ventricles. The pulmonary and aortic, a pulmonary artery and aorta. So that's what's stopping them. A couple more questions then. Let's have another go. How does blood leave the left side of the heart? And what does the right side of the heart do? And why is that muscle so much thicker on the left side of the heart? Okay, I'll put those answers up then. So blood's leaving the left side of the heart on the aorta, through the aorta. And what does the right side do? Well, the right side is receiving the deoxygenated blood, so it takes it to the lungs. And the left side is much thicker because it has to pump blood further all the way around the body than just to the lungs. So 100% there for praise. Excellent. Really impressive, especially without the images. So what we're going to talk about next is the fetal circulation, because obviously we're getting, as adults or just as children or babies even that have been born, we're all getting our oxygen from our lungs. And if you're in utero, so in the uterus, you can't breathe because you would be just inhaling the fluid there. So we've obviously need a slightly different circulatory system. So we need to start looking at the placenta over here. Now the placenta is another exchange surface. So we talked about the lungs being an exchange surface. We've had quite a good look at the small intestine being an exchange surface. And now this is another exchange surface. This is the placenta. So common features of exchange surfaces are large surface area. We had a look at the adaptations, those villi, to increase the surface area of the small intestine. They have a concentration gradient because the difference in concentration in ions will drive movement of particles. It doesn't have to be ions, it can be any particle, but it will move particles. Remember, particles move from a region of their high concentration to a region of their low concentration. So if you haven't got much of a difference, they won't move. If you've got a concentration difference, they will move high to low concentration. So large surface area, concentration gradient, that's maintained by a really good blood supply. A short diffusion distance. So they're normally only about 1 cell thick. And if they're moist, but most surfaces in the body are moist as well. Right, so through these blood vessels in the umbilical cord, the fetus will be getting the nutrients from the mother's blood and oxygen, which the mother has inhaled as well. Waste products that are made in the fetus, so the fetal cells are using up the oxygen and the nutrients, will get sent back through and then removed by the mother's body. So the mother will breathe out the CO2 that the fetus is producing, for example. The mother's blood does not move into the baby. So the mother's blood is obviously carrying oxygen, but it's carried attached to the blood vessels, the red blood vessels, but they don't move into the fetus. Those red blood cells don't move, just the oxygen goes. So because the fetus hasn't got lungs, it uses 3 shunts, which are small passages that direct the blood that need to be oxygenated, so it doesn't go into the lungs of the fetus. I've just got to move a little box on my screen again. And what they do is they stop that blood from going to the lungs and to the liver until after the fetus is born, and they'll move the blood from the right atrium into the left atrium of the heart. So remember when we were looking at the adult heart, I was telling you there's a septum that divides them. There's 2 hearts and the 2 parts of the heart, 2 pumps, and they don't mix. However, in the fetus, the blood will cross. It can move directly from the right atrium to the left atrium through a little hole. And that can sometimes occasionally stay open after birth. You might have heard of people talking about babies having a hole in the heart. That's what happens. So these little ducts then, we have the ductus arteriosus, which moves blood from the pulmonary artery to the aorta. Oxygen nutrients from the mother's blood are sent across the placenta to the fetus, as we know, and this is enriched with everything that the baby needs. Well, fetus, we should say, it's not really a baby yet. Then When the blood reaches the inferior vena cava, it is sent through the ductus venosus, and this shunt lets highly oxygenated blood bypass the liver directly to the vena cava and then to the right atrium of the heart. So a small amount of this blood goes straight to the liver to give it oxygen and nutrients that it needs to develop and grow. And then the waste products are sent back to the mother's blood. So rather than doing the loops that we would expect, because it can't go to the liver and to the lungs in the fetus, we have these two ducts moving the blood. Ductus arteriosus, so from pulmonary artery to the aorta, so arteriosus meaning arteries, and the ductus venosus there, moving blood to the vena cava. So, as I was saying a moment ago, oxygen is carried on the red blood cells. This is because oxygen has a really low solubility in water, and blood is mostly water. So that plasma that you have, you'll have seen it when I mentioned a couple of sessions ago, when you used to get a cut when you were little, if you fell over and actually grazed your knee, you'd see that sort of yellowy fluid, that's the plasma. That is what all of these cells are suspended in, and it's really rubbish at carrying oxygen. It has a really low solubility. So blood has adapted to carry oxygen in specialized molecules called hemoglobin, and these are found in the red blood cells. So red blood cells have adapted for their function. They're really highly specialized cells, and usually if somebody is asked in an exam question to give an example of a specialized cell, this is probably one of the ones that comes to mind straight away, because it's one you learn about first at GCSE level. So it's, it's considered quite an easy one. So red blood cells have no nucleus in their adult stage, I hasten to add, but you don't write that. Everyone just says red blood cells have got no nucleus, so they can carry more oxygen. There's more space inside them to carry oxygen. They don't live very long. Only a matter of weeks. Some cells last for ages, years. Red blood cells just only last a couple of weeks because they've got no DNA in their adult form, because they have this lacking a nucleus situation. So they can't carry out repair when damage takes place. They are a biconcave shape. That's the shape you can see there. It's a bit like a donut, but without the middle missing. And the reason it's got that biconcave shape is it increases the surface area to allow the oxygen to diffuse in to the cell. And as I said, it's got these specialized hemoglobin proteins inside. They're made up of 4 subunits, so 4 chains stuck together of amino acids, and they've got a heme group in the middle, and that's just something that contains an ion, that says atom there, but it is actually charged, so it's an ion. And what that ion does is it binds to molecular oxygen. And each hemoglobin molecule can carry 4 molecules of oxygen. And this is what is going on in the lungs. It's picking up the oxygen. It passes out to the rest of the body, releasing the oxygen to the cells. And then the oxygen is being unloaded by the red blood cells, and they can also absorb some carbon dioxide from the tissues. Not all of the carbon dioxide is carried in the red blood cells. Some of it's carried in the plasma, and the carbon dioxide isn't carried by this specialized hemoglobin molecule. The hemoglobin is just for oxygen. Some of it, 20%, because otherwise it would affect our ability to carry oxygen, and we don't want that competition or we'll die. And that's actually how carbon monoxide kills you. Carbon monoxide, which instead of being CO2 is just CO, so that's carbon dioxide, CO2, and if you've just got one oxygen, it's carbon monoxide. That actually competes with the oxygen and will kill you, but the carbon dioxide doesn't compete. I mean, 20% does, I suppose, but doesn't massively compete with the oxygen. So we can still carry oxygen if there's CO2 around. So what it does is binds to the protein bit. It doesn't bind to the actual heme bit, so it's not a problem with transport. So, a couple of questions then. These are just relating to specialized blood cells. Where's the hemoglobin picking up the oxygen? What proportion of CO2 is carried by hemoglobin? And what is the primary function of phagocytes? Bit of a flashback to last lesson there. It's a little bit easier than the earlier questions, I think. Let's have a look. So hemoglobin's picking up the oxygen in the lungs, but 20% of CO2 is carried by hemoglobin, but not competing with the oxygen. It is on the molecule, but nothing significant to worry about. The oxygen binds to the ion, so no massive competition. What's the main component of blood that carries the oxygen? What type of blood cell is primarily responsible for oxygen transport? Have you remembered the proper names there for the blood cells? That's a tricky one. And what shape are the red blood cells? Okay, I'll put those up. So, is the red blood cells or erythrocytes that carry oxygen? So that's the proper name for the red blood cells. And they are specialized, adapted to their function by being biconcave in shape, which gives them a large surface area for the oxygen to diffuse in, and also having no nucleus, so they can fit extra. I'm going to skip through that one for now. And they've got no nucleus, so they can fit extra oxygen in. So let's have a look at hemoglobin now. So the fetus has hemoglobin, which is slightly different to the hemoglobin that we have. So we were just mentioning hemoglobin molecule, iron atom, ion technically. I'll let your chemistry teacher explain that in the middle, is the oxygen transporting protein in our red blood cells. Now, it has 4 subunits, and they're actually handily coloured in different colours on this diagram. So you can see it's got 2 blue ones, 2 red ones. We won't worry about the names. It's got 4 subunits and a heme group containing iron in the middle. This is it. So Fe is the symbol for iron, and it binds to oxygen. So that's what it looks like. The fetus has a slightly different type of hemoglobin compared to the one that is found in adults. So we call them HbF and HbA, so the hemoglobin fetal and hemoglobin adult. So the fetal hemoglobin is the main oxygen transport for the fetus during the last 7 months of its development in the uterus, and it is still in the body as a neonate until, so new baby, until roughly 2 to 4 months old. And it differs to adult hemoglobin in that it will grab onto oxygen better. So the oxygen will bind with a greater affinity than the adult form. So what we're doing is essentially giving the hemoglobin in the fetus a much better affinity for the oxygen, so it will grab it off the mother. So let's have a little look at some of these components of the blood then, since we've talked about the the red blood cells quite a bit. Let's talk about everything else. So the fluid plasma is the yellowy fluid that I was talking about that you see when you've got a graze rather than a cut. And several components circulate within this plasma. There's the red blood cells, which we've talked about quite a bit. They're properly called erythrocytes. Loads of different white blood cells that we've talked about already. We've talked about phagocytes and lymphocytes quite a bit. And then there are also platelets. Now, these aren't technically cells, but they're bits of cells that have been pinched off. They're bits of cell cytoplasm. And we'll talk about them when we talk about clotting. We've also got loads of hormones circulating. Those are really important communication chemicals. And then we've got all the digestive foods, such as glucose, the nutrients that we need. And we've got the waste materials that we talked about when we were discussing the liver, and the CO2 that we get when our cells are respiring and using up that oxygen. And the blood circulates through the body, through all of the tubes, some of which we talked about with the heart, then the arteries, the veins, and the capillaries. So let's have a little look at the content of the blood then. So about half of it is blood. So the red blood cells, white blood cells, which are involved in defense, and the platelets, which are technically not full cells, they're just bits of cells. There are loads of different white blood cells. We've talked about phagocytes that ingest microorganisms, and we did talk about some lymphocytes a little bit last session. We talked about some T cells. Haven't talked about the B cells yet, but we will. So that's the blood cells, but there's so much stuff going on with this plasma. It's nearly all water, which is why our blood is a liquid, but there are 8% dissolved substances, hormones, foods such as glucose and amino acids and fats and stuff. There are some salts, there are proteins, and we are going to learn about fibrinogen, which is used for clotting. There's also the waste products that we've mentioned, and then there are other proteins like albumin, which is a liver protein, which is important for maintaining the pressure, osmotic pressure of our blood. So all of the components of the blood, excluding the red blood cells, the erythrocytes, can leave the blood vessels via the capillaries. So once the blood has been pumped out of the heart, it goes into the aorta, and then that takes it all around the body. It's full of oxygen. At this point, remember, the red blood cells are carrying the oxygen attached to this hemoglobin. And when it gets from the arteries, it goes into slightly smaller arteries type vessels. They're called arterioles, and then it ends up in the capillaries. And the capillaries are very, very thin. They're only 1 cell thick, and all of the components of the blood, except the red blood cells, can leave, and they can just go off into the tissues, off to the cells. So at this point, it's no longer called blood, but instead it's called tissue fluid, or interstitial fluid is the proper name, but tissue fluid, everyone knows what that means. It includes white blood cells and lymphocytes. These can literally squeeze between the cells and get out of the capillaries and into the tissues. The platelets can get into the tissues. Everything that's dissolved, the hormones, the food, all of the waste materials can all get in and out of the tissues. And let me just check how we're doing time-wise. Okay. We're going to have a little look how some of those substances that are in the plasma do their job. We're going to have a look at blood clotting. So if a blood vessel is damaged and the blood comes into contact with new surfaces, 2 things happen. The platelets become activated, and the fibrinogen, which is a protein which is floating around, which is soluble, becomes insoluble. It turns into its insoluble form called fibrin. So it's chemical reactions make that happen. What happens when the platelets are activated is they begin to stick to each other, and they stick to surfaces. So they clump, and they clump to the vessels as well. And that fibrin, which is now insoluble, makes sort of fibers. It looks like fibers, and these trap the blood cells. So you have fibers, platelets, and blood cells all trapping together, and they can plug injured, damaged blood vessels. So as we said, the platelets are these little bits of cells. Another thing that will happen when all of these processes are going on is they will cause the blood vessels depending on their size. They have muscle in them. Not so the capillaries don't have muscle in them. They're way too small, but the arterioles and arteries have muscles in them, and it will contract, squeeze together to stop the blood leaving it, really. And you'll have platelets forming the plug. This is reinforced by the fibrin. This traps the blood cells and forms a clot. Platelets are, everything is accumulating and sticking together, reinforced and tangled together by these fibrin particles and stopping you bleeding out, essentially. So that's how your clotting process works. So here we've got something that you need to cover. This is called an oxygen dissociation curve. And what it shows is the percentage of hemoglobin saturated with oxygen on the y-axis, which is the one that goes up, and the partial pressure of oxygen on the x-axis. So oxygen has a higher affinity, hemoglobin, sorry, has a higher affinity for oxygen at high partial pressures. And that's the sort of thing we'll find in the lungs, where it will easily bind to oxygen, and it has a lower affinity or ability to hold on to it at low partial pressures, and that's what we'll find in the tissues. So that's good. That forces oxygen to be picked up in the lungs, and it's much easier to lose it in the tissues, which is exactly what we want because of the difference in pressure. We get this S-shaped curve, which is called a sigmoid curve or sigmoidal. And that's due to the fact where something called cooperative binding happens. The first oxygen molecule makes it easier for the next oxygen molecule to join. So once one has joined, it's easier for the next one to join. So it sort of increases the rate, it speeds up. It'll be a bit slow to start with, there's a bit of a lag, then it speeds up. So let's have a look at some important points on this curve. So the flat upper part is the oxygen loading zone. So even if the pressure drops slightly, saturation stays high, and that means there's stable oxygen loading. And then the steep part of the slope is the oxygen unloading zone. And we get something called the Bohr effect. What this explains or describes is probably a better word, is the changes in carbon dioxide concentration and pH and how they affect hemoglobin's affinity, so how much it wants to stick to the oxygen. How are we doing time-wise? Okay. So when the tissues produce more carbon dioxide and acid, it lowers the pH. So acid in the blood, carbon dioxide in the blood changes the pH, and it does make it acidic, a bit acidic. And when the tissues produce more CO2, essentially there's more acid around. So the hemoglobin will release oxygen more easily because there's more CO2 to be removed. So hemoglobin's ability to bind to the oxygen depends on where it is in the body, so it depends on the environment. So CO2 concentration, hydrogen ion concentration, so that's how acidic it's getting, but that's caused by CO2, essentially, one of the reasons. And when it becomes more acidic, the oxygen's the hemoglobin's affinity for the oxygen will decrease, and you can see that the oxygen curve, the graph, essentially will move to the right. So oxygen is released more easily in the body tissues. So the more CO2 it is, the more acidic it is, the more easily the oxygen is released, and then carbon dioxide will be picked up as well. And that makes more sense because it's more acidic and more CO2 in the tissues, which have been doing their job, but they've been using oxygen up for respiration. So that means they're going to need more oxygen. They produce in the CO2. So that just makes it easier for the hemoglobin to sort of know when to get rid of the oxygen. And then you can see here the fetal hemoglobin and adult hemoglobin. Remember, we said that it differs slightly, and it's easier to bind to the oxygen. There's greater affinity to oxygen than the adult hemoglobin. So that's what that looks like on a curve as well. Okay, we are pretty much at the end of time. So if you've got any questions, you can let me know and I can get back to you on them if they're long, or let you know the answer if they're short. And we will continue with the theme next session. Just pop my chat up so I can actually see. But if you've got no questions, have a good rest of your day and I will see you on Tuesday. Bye guys. Bye-bye.